Hello, aspirants. Welcome to another high-yield explainer for your GS Paper 2 preparation, brought to you by Rungta IAS Academy. Today, we are discussing a massive paradigm shift in reproductive public health, paternal epigenetics.
Exactly, sir. We're diving straight into a topic that completely challenges how we traditionally view our national health missions. It's a truly brilliant intersection of cutting-edge science and governance.
Now look at this highly probable prompt for your upcoming UPSC mains examination. Critically examine the focus of India's reproductive health interventions. Why are fathers missing? This right here is the exact analytical lens we'll be applying throughout today's session.
To tackle that question, we first need to evaluate the status quo. You see, India's RMNCH plus A program has undeniably succeeded in bringing down maternal, neonatal, and childhood mortality over the last 3 decades. It's a huge achievement. But a quieter, much more serious concern is surfacing. Children are increasingly vulnerable to metabolic disturbances, recurrent infections, and frankly, a lack of physical robustness. We've focused so heavily on just survival, but the biological quality of that survival, that is now raising some serious alarms.
So, why exactly is this happening? Well, for over a century, the traditional mother-centric model, which was heavily reinforced by August Weismann's blood-testes barrier concept, taught us that sperm only passively transmitted DNA. We thought it was completely insulated from the father's external environment. But the emerging scientific model shifts us to an inclusive, biparental understanding. It turns out a father's environment actively shapes the child's biology long before conception even happens. Now, for our aspirants coming from a non-medical background, let's break this complex science down simply. Think of DNA as the hardware of a computer, right? And epigenetics is the software. A father's diet, his stress levels, and his exposure to toxins, they don't actually change the genetic code, the hardware itself. Instead, these traits are transmitted like software updates, specifically through small RNA fragments called sperm microRNAs. These tiny molecules actually regulate gene expression in the early embryo. Let's look at some hard data that brilliantly illustrates this. This is from a recent study published in the journal Cell Metabolism. They took male mice and put them on an 8-week structured treadmill exercise routine. The result? Their offspring showed vastly improved endurance. Quantitatively speaking, the offspring were running 30 to 40% greater distances. Muscle analysis even showed a physical shift towards oxidative fibers, so the father's lifestyle directly programmed the next generation's physical robustness. Absolutely fascinating. And at the molecular level, the mechanism is quite elegant. During fertilization, these sperm microRNAs are acting as active agents, not just passive markers. When they're delivered to the oocyte, they suppress a specific molecule called nuclear receptor corepressor 1. By inhibiting this corepressor, it essentially unblocks a master regulator. This directly programs the embryo for enhanced oxidative metabolism and mitochondrial development right from the very start. What this ultimately means, and aspirants, please note, this is a fantastic, high-scoring point to drop into your mains answer, is that the classical Weismann barrier is actually selectively permeable. It totally bypasses traditional genetic insulation. These circulating RNA fragments act as molecular signals from the father's somatic tissues, directly influencing the germ cells. They are reprogramming gene expression without ever altering the fundamental DNA base pairs. So, let's connect this cutting-edge science directly back to your GS Paper 2 governance syllabus. Let's analyze the policy gap here, drawing from the insights of public health physician Dr. C. Aravinda. If you look at our RMNCH plus A program, the focus is almost entirely on antenatal care, maternal nutrition, and institutional deliveries. What about male involvement? Well, it's severely limited to basically just handing out iron tablets to adolescent boys. There is a complete absence of any structured focus on paternal preconception health, no systematic screening for environmental and lifestyle risks among prospective fathers whatsoever. Which brings us to the way forward for your essay or your answer conclusion. Addressing paternal lifestyle factors, things like physical activity, substance use, and environmental exposures among men of reproductive age, this offers a highly effective, low-cost, high-impact public health strategy. If these animal model findings hold true in humans, the implications for improving our entire population's health across generations are just profound. Finally, let's test your administrative aptitude. Here is a provocative question for your personality test and interview board preparation. If appointed as health secretary, how would you integrate paternal lifestyle screening into national health policy? Really think about it, aspirants. How do you apply this advanced science to grassroots governance without alienating the population? Keep analyzing, keep interlinking those syllabus topics, and we will see you in the next explainer right here at Rungta IAS Academy.